professor, physician, epidemiologist. I am Dr. Sri Banerjee.
Epigenetics and public health. This is Dr. Michael Schwab. I'm on the core faculty in the School of Health Sciences at Walden University.
And I am Dr. Sri Banerjee, and I am core faculty for College of Health Sciences.
So our first slide, objectives of this presentation. First, we want to look at how epigenetics is different from genetics. And second, we will articulate the basic concepts of epigenetics. And finally, explain the role of epigenetics in social factors contributing to public health. There are some key terms in genetics that we need to look at. Genes. Genes hold the instructions for building proteins needed for cells to function. They're made up of DNA, deoxyribonucleic acid, in about 3 billion base pairs. Genetics is the study of genes and their role in the inheritance of physical and mental traits. Then genomics. This is the study of the whole genome, the complete assembly of genes. And finally, we'll be referring to this term omics, which is the sciences that aim to categorize wholes rather than parts.
In contrast, we will also be looking at the concept of epigenetics. Now note, epigenetics, even though the term genetics is included within this, is distinct from genetics. Epigenetics can generally be contextualized as the study of heritable, non-genetic chemical compounds. And as we discuss this later on, you'll see that a lot of these may be derived from parental adaptations and behavior. And what makes this all the more influential is that this has the potential to influence the expression of genes in the offspring without altering the primary genetic code of the individual. Epigenomics is actually the study of the aggregation of compounds that attach to genes. Extending from our prior definition of omics, when you add this ending to any of these fields, then automatically you are thinking in terms of a whole rather than in a more reductionist sort of specific approach. Finally, epigenomic compounds are said to, quote, tag or mark the genome. So simply put, a mark is a change in the way the cells use the genetic instructions. So again, the genetic code remains the same. However, when it comes to expression of these traits, of these inheritable traits, those are altered by these compounds. In this next visual, this becomes even more self-evident. As you can see, the chromosome is the unit in which all of these smaller groups are packed. So in a normal, in an average human, the number of chromosomes are 46. We have 23 pairs of chromosomes. So in all of these chromosomes are encoded very specific genetic material, which are comprised of 4 base pairs. So if you look at that chromosome and then build it up and then take it apart into smaller groups. You think about the chromatin, you think about the DNA that comprises the chromatin, and then these methyl groups that are actually causing alterations in expression.
Okay, so that's the techniques, the mechanism of epigenetics. And now we go to, well, we're so used to thinking of genetics as determining our behavior and our health, but there's an ancient debate, it goes back over 100 years to the beginning of genetics, a debate about whether it is the genes or whether it's our environment, upbringing, and life experience that determine our behavior and our health. It's an old debate by now, and we've come to a place of recognizing that we know it's both. Not only the genes that you inherit, but also the epigenes that you inherit, which regulate how, to some degree anyway, how the genes are expressed. Now, on the next slide, we look at how epigenes, epigenomics, relates to social factors. Social factors being the many social and environmental determinants of health. The epigenomic, and you say now epigenomic rather than epigenetic because we're looking at the whole genome, the epigenomic inheritance of parental adaptations and behaviors, like parents' and ancestors' responses to the trauma of slavery, may affect the health of offspring over generations. The effect of this epigenomic inheritance can operate through social factors. For example, the ways that we live in our social and environmental context, and that means that they may operate through health or education, access to education, income, and access to healthcare. This can happen in some ethnic and racial groups, even when we control for social factors, which leads us to believe that epigenomic factors may also have an independent activity, regardless of social conditions. We use this scenario to look at how epigenomics may affect the higher infant mortality rate for Blacks than for whites in the US today.
As we delve further into looking at some of these disparities, specifically as we are discussing about race, these figures show very distinctly some of these disparities. So if you start with looking at the table on the left-hand side, as Dr. Schwab was mentioning, there are disparities that you see between Blacks and whites. So as it pertains to everything from cancer, everything from cardiovascular disease, and other areas like homicide, there's distinct disparities. And if you look at the number at the very right column, which says Black-white ratio, the higher the number, the more disparate the findings are. So the higher the number, the higher the disparity between Black individuals and white individuals. In order to bring to light the issue of infant mortality, the figure to the right clearly indicates that the infant mortality rate per 1,000 live births. Now, this is key. When you're thinking about certain rates, certain mortality rates, when you're reporting infant mortality rates, these are always reported per 1,000 live births. So as you're thinking of units, please keep this in mind. But to the larger point, if you see the distribution, non-Hispanic Blacks experience the highest rates of infant mortality. And these disparities are striking. As we continue to understand and articulate these disparities, there is a need for theoretical underpinnings to better understand these social factors. So there are currently 4 broad versions of the social determinants of health. The World Health Organization initially in 2010 had a robust model explaining social determinants of health, but subsequently, because this is such an important topic, various organizations and agencies define this from slightly different perspectives. So County Health Rankings created a model. Kaiser Family Foundation in 2018 created another model. And then finally, the most recent is the Healthy People 2030, which the individuals from the government define social determinants of health. So here in the next slide and here, we will be talking predominantly about the most recent social determinants of health update. And broadly speaking, this is divided into 5 categories. The categories are education access and quality, economic stability, social and community context, neighborhood and built environment, and healthcare access and quality. Each one of these, I can give you multiple examples of how these social determinants affect overall health. Thinking back through the COVID-19 pandemic and massive shutdowns globally of public conveyances and of businesses, this led to economic instability. And furthermore, some of this economic instability led to worsening issues arising from the pandemic. Similarly, we can use the COVID-19 pandemic to illustrate some of these other social determinants. So if you can take a moment to think about examples, suitable examples of each of these social determinants, then this can help you understand these concepts better. So what is social epigenomics? Since we have defined now what some of these social factors are, what some of these social determinants of health are, now it's important to bring all of these concepts together. How does social determinants of health impact epigenomics? And so if we look at this visual, we see the cause and we see the effect, but it's important to articulate some of these reasons, which will be done in the subsequent slide. But very importantly here, it's important to note that race and ethnicity determine social environment and social experiences, which affects gene function at various stages of life, and eventually thereby influence health trajectory and modify disease risk.
So that's the big picture of the role of epigenetics in public health. And we conclude by with these three proposals that we inherit genetically and epigenetically, and that both influence the expression of genes. Epigenetics can be just as important as genetics, as we've seen in the example of Black infant mortality in this country. And epigenetics can serve as one of the explanations for health disparities through a wide range of diseases, either through social factors or independently. And that, my friend, is that.
Thank you for listening.